Hello dear students, welcome back to Biotechnology. I hope you all are doing very well. Today in this lecture video we are going to discuss about one important topic from molecular biology, that is histone modification. This is very important topic for various competitive examinations in life science like CSIR NET, GATE, DBT, ICMR, etc. So today we are going to discuss about the histone modifications such as acetylation, methylation and phosphorylation. So without further delay, let's get into today's class. As we know that our chromosomes are made up of nucleosome. Here you can see that nucleosome. Okay. And it is like beads on a string-like structure. And the nucleosome has two components. One is histone core. Here you can see that histone core. This blue color ones are histone protein. And second is DNA. This string, red color string is DNA. Okay. And DNA is wrapped around this histone core, making the beads on string-like form. Okay. Inside this histone core, there is different types of histone protein like H3, H4, H2A, H2B. See, here in this picture we can see that 8 histone protein. Core of 8 histone molecule H2A, H2B, H3 and H4. Here there is a linker histone, that is H1 histone. This is H1 histone. Okay. And DNA. This blue color one is DNA. DNA is wrapped around this histone octamer. Okay. We know that DNA has negative charge. DNA has negative charge. And histone has positive charge. Histone has positive charge, because it contains a very high amount of positively charged amino acids such as lysine and arginine. That's why histone has positive charge. We know that opposite charge will attract each other. So, because of this opposite charge, DNA is wrapped around the histone core. Okay. Because of this positive charge only, DNA is wrapped around the histone core. And histone modification cause some impact on histone DNA interaction. Some modification disrupt histone DNA interaction. See here. This is histone octamer and this is DNA. Okay. After histone modification, that will change the interaction of histone with the DNA and causing nucleosome to unwind. Here you can see that loosening of DNA. Okay. And it will cause open chromatin formation and it is called the euchromatin. Here in this picture you can see that this is open chromatin and that is called the euchromatin, and that leads to gene activation. Okay. And some modification strengthen histone DNA interaction and that leads to heterochromatin formation, and that leads to gene silencing. Gene silencing. So, in this way, histone modification change chromatin structure and that leads to gene activation or silencing, or we can say that histone modification will lead to repression or stimulation of gene expression. Okay. So some histone modification leads to Activation of the gene and some histone modification leads to inactivation of the gene. That means silencing of the gene. And this histone modifications includes acetylation, methylation, phosphorylation, ubiquitination, ADP ribosylation. So these are some histone modification. Here we are going to discuss about acetylation, methylation and phosphorylation. Okay. Let's start with acetylation. What is meant by acetylation? Acetylation means it is the addition of acetyl groups to lysine residues in the N-terminal ends of the histone. We know that proteins have C-terminal and N-terminal ends. C-terminal and N-terminal ends. Okay. And histones are also proteins. So, histone protein contains 11 to 37 amino acids in the N-terminal end. And this N-terminal end is called the histone tail. This N-terminal end is called the histone tail. See, here you can see that this is histone protein H3 and this is histone tail. Okay. So, N-terminal end of histone is called the histone tail. Acetyl groups are added to the lysine residue that is present in the histone tail. Acetyl groups are added to the lysine residue present in the histone tail. Okay. So, acetyl groups are added to the lysine residue of histone by histone acetyl transferase. Histone acetyl transferase. Short form is HAT. Okay. Here you can see that acetyl group. So acetyl groups are added to histone tail by histone acetyl transferase, and this is reversible process. So, and this acetyl groups are removed by deacetylation by the enzyme called the histone deacetylase. Histone deacetylase. Okay, and short form is HTACS. So, acetyl groups are added to lysine residue of histone by histone acetyl transferase, and it is removed by histone deacetylase. Okay. You have to remember that acetylation will lead to activation of the gene. Acetylation will lead to activation of the gene. Okay. And deacetylation. Deacetylation will lead to inactivation of the gene. Okay. Unmodified lysine residues are positively charged. Here you can see that positively charged. Okay. So unmodified lysine residues are positively charged, but acetylation result in the neutralization of the charge, that will lead to the neutralization of the charge on histone, and which reduces the interaction of histones and negatively charged DNA. Okay. So the charge neutralization result in a weaker histone DNA interaction and allows transcription factor binding and gene expression. Okay. After acetylation, what will happen? Bromodomain containing proteins will come. This is bromodomain containing protein. Bromodomain containing proteins recognize histone modification and it binds with acetylated lysine in the histone tail, and that can change the chromatin to active state. So transcription factor can come and bind with that. Okay. So after acetylation, bromodomain containing proteins will recognize this modification and it will bind with acetylated lysine, and then it will change the chromatin to active form. Okay. And you have to remember that heterochromatins are inactive, but euchromatins are active, isn't it? So histones in heterochromatins are generally deacetylated, whereas euchromatins are acetylated. Okay. So you have to remember that acetyl groups are added to lysine residue of histone tail of the core molecule. Unmodified lysine residues are positively charged, and acetylation leads to neutralization of the charge. So what will happen? It will result in the weaker histone DNA interaction. And histones in the heterochromatins are deacetylated, whereas euchromatins are acetylated. Okay. Example for acetylation: H3K9. H3K9 means acetylation of K9. K9 means 9th position lysine on histone H3 and 27th position lysine of histone H3. This will lead to enhanced and promoter gene activation. Okay. So acetylation will lead to activation of the gene. That is the main thing. Okay. Methylation is the addition of methyl group to lysine or arginine residues of histones, mainly histone H3 and H4. Okay. Histone methylation is catalyzed by the enzyme called the histone methyl transferase. Histone methyl transferase. Here you can see that methyl group. Okay, so histone methylation is catalyzed by the enzyme called histone methyl transferase HMTS. And it is also a reversible process. So histone demethylase that will remove this modification. Histone demethylase HDMS. It will remove methyl group. Okay. But this histone methylation does not alter the charge of the histone protein. As we studied, the acetylation will lead to the neutralization of the charge. But here, this methylation that does not alter the charge of the histone protein. This is very important. Okay. And histone lysine methyl transferase. Histone lysine methyl transferase HKMTS. HKMTS. It will methylate lysine residue. Okay. It will methylate lysine residue and protein arginine methyl transferase PRMTS. It will methylate arginine residue. Here it is given. Histone lysine transferase HKMTS methylate lysine residue protein arginine methyl transferase that will methylate arginine residue. Okay. Unlike acetylation, methylation is associated with both activation and repression. We know that acetylation will lead to activation of the gene, but here methylation is associated with both activation and repression depending on the amino acid and position of modification. You have to remember that arginine methylation promote activation while lysine methylation that is associated with both activation and repression. That is depending on the methylation site and degree of methylation. Okay, arginine methylation promote transcriptional activation while lysine methylation is implicated in both transcriptional activation and repression depending on the methylation site. Okay. For lysine, mono, di or trimethylation is there, but for arginine, only mono and di methylation is there. Okay. So, mono, di or tri for lysine, I will write here mono, di, tri for lysine and mono and di for arginine. Okay. Here you can see that this is lysine and here one methyl group is there. This is monomethyl lysine, monomethylation. Here two methyl groups are there and here dimethyl lysine. And here three methyl groups, so this is trimethyl lysine. But for arginine only mono and di. Okay. For example, methylation of lysine at the 9th position of H3. I will write here methylation of lysine at the 9th position of H3. That means H3K9. Okay, H3K9. It is associated with gene silencing. Gene silencing. That means repression. Okay, and H3K4. H3K4 is associated with activation. And H327, dimethylation of H327 and H3K9. Dimethylation of, here I am writing, this is important. Okay. Dimethylation of H3K27 and H3K9 both will lead to repression. That means silencing. Okay. As I told you that acetylation of these two H3K27 and H3K9 will lead to activation, but methylation, that means dimethylation of H3K27 and H3K9 will lead to repression, that means silencing of the gene. This is important. After methylation, what will happen? This methylated lysine or arginine that will provide binding site for proteins containing domain called the chromodomain. Okay, I will draw here. This methylated histone will provide binding site for proteins with chromodomain containing protein. Okay, which then leads to the alteration of chromatin structure. Okay, As I told you, acetylation provide binding site for bromodomain containing protein. Here it is chromodomain containing protein. Okay. So that's all about methylation. Next is phosphorylation. Phosphorylation means it is the addition of phosphate group. It is the addition of phosphate group. Phosphorylation takes place on all histone proteins with a different effect on each. Histone phosphorylation mostly occurs on serine and threonine residues but also occurs on tyrosine residues. Okay, this is very important. You have to remember this. Histone phosphorylation takes place on serine, threonine and tyrosine residues. Okay. And protein kinases. Protein kinases add phosphate group to histone tail. And this will lead to gene activation. Phosphorylation will lead to gene activation. Okay. While dephosphorylation, that means removal of phosphate, will lead to gene suppression. That means repression of the gene. Okay. So who is removing or which enzyme is removing phosphate group? Phosphatases. Phosphatases is removing phosphate group and this will lead to a repression of the gene. Repression of the gene. Okay. So protein kinases catalyze the binding of phosphate group to the amino acid at the tail of the histone, which is involved in transcriptional activation of the gene, and protein phosphatases on the other hand removes phosphate group from the amino acid residue and suppress the initiation and expression of gene transcription. This is very important. Phosphorylation will lead to gene activation. Dephosphorylation will lead to gene suppression. Okay. So the enzyme protein kinase that will transfer phosphate group from ATP to the hydroxyl group of the target amino acid like serine, threonine and tyrosine. Here I will draw the structure of serine. Here NH3, CH3 and here hydroxyl group. Okay. So protein kinase that will transfer phosphate group from ATP to hydroxyl group of the target amino acid. Okay. So here only phosphate group will come. Okay. Here you can see that dephosphorylation, removal of phosphate by protein phosphatase. So like acetylation, phosphorylation is generally associated with transcriptional activation and phosphate group is negatively charged. Phosphate group is negatively charged. If we add this phosphate group to the amino acid, a repulsive force will create in between histone and DNA. That means histone DNA interaction will reduce and that will lead to the activation of the gene. Same like acetylation. Okay. Example for phosphorylation. Serine 10 of histone 3. That means H3S10 phosphorylation during mitosis by Aurora B kinase. Okay. This will lead to gene activation. Serine 10 of histone H3 during mitosis by aurora B kinase is an example for phosphorylation, histone phosphorylation. And histone phosphorylation plays important role in mitosis, cell death, DNA damage repair, DNA replication, etc. Okay. So that's all about phosphorylation. I think this is clear for you. If you have any doubts, then please ask me in the comment section. I will try to reply your comments. So here I am winding up this lecture video. If you like this video and find it helpful, then please click the like button, share this video with your friends, and also don't forget to check whether you subscribe this channel or not. If you didn't subscribe, then please subscribe right now and turn the bell icon to all. Keep learning. Thank you.